Okay, chapter 10, meiosis. Living organisms are distinguished by their ability to reproduce their own kind. Heredity is the transmission of traits from one generation to the next. Variation is demonstrated by the differences in appearance that offspring show from parents and siblings, and genetics is the scientific study of heredity and variation. In a literal sense, children do not inherit particular physical traits from their parents. What they do inherit are genes, which are units of heredity that are made up of segments of DNA. Genes are passed to the next generation via the reproductive cells called gametes, which are your egg and sperm. Most DNA is packaged into chromosomes. For humans, we have 46 chromosomes in our somatic or our body cells. In our gametes, we have 23. So 46 would be the diploid number, and 23 would be the haploid number. Each gene has a specific position or location or locus on a certain chromosome. In asexual reproduction, a single individual passes its genes off to its offspring without the fusion of gametes, so there is no genetic variation when there is asexual reproduction. A clone is a group of genetically identical individuals from the same plant. In sexual reproduction, however, you have two parents that give rise to offspring that have genetic variation, and they are unique combinations of genes from both parents. Life cycle is the generation-to-generation sequence of stages in the reproductive history of an organism. Human somatic cells have 23 pairs of chromosomes, or 46 total. A karyotype is basically a picture of those chromosomes. And you can see the homologous chromosomes that are coming up and pairing together. So that looks like this right here. So this is a metaphase chromosome. This is when the chromosomes line up at the equator, so you can see the homologous chromosomes lining up. So this is a pair of duplicated chromosomes, this is a duplicated chromosome, and this is a second duplicated chromosome. Okay, because remember in meiosis 1, your homologous pairs come together. Meiosis 1, metaphase 1, the pairs line up at the equator, and that's when this picture gets taken, when they're lined up together. So you can see all of the homologous pairs of chromosomes lined up. You can see that there's 1 through 22, and then you can see that there's an X and a Y. The sex chromosomes, which determine the sex of the individual, are called X and Y. Human females have a homologous pair of X chromosomes, so females are XX, males are XY. The remaining 22 pairs of chromosomes are called the autosomes. Each pair of homologous chromosomes includes one chromosome from each parent. The 46 chromosomes in a human somatic cell are 2 sets of 23. You get 1 set from your mother and 1 set from your father. A diploid cell, or 2N, has 2 sets of chromosomes, and in humans that's 46. In a cell in which DNA synthesis has occurred, each chromosome is replicated. Each replicated chromosome consists of 2 identical sister chromatids. So these are homologous chromosomes. So you can see the blue represents the one that came from the dad, and the red ones represent the maternal chromosomes, or the one that came from mom. So notice that homologous chromosomes are pretty much the same size. So this would be a pair of homologous chromosomes, this would be a second pair, and this would be a third pair. So they are similar in shape, size, and they contain the same genes, different forms of those genes. A gamete contains a single set of chromosomes and is haploid. For humans, the haploid number is 23. Each set of 23 consists of 22 autosomes and a single sex chromosome. In an unfertilized egg or an ovum, the sex chromosome is X, and in a sperm cell, the sex chromosome may be either X or Y. Because of that, it's actually the male that will determine the sex of the offspring, because the female can only give an X, whereas the male can give an X or a Y. Fertilization is the union of gametes. The sperm fertilizes the egg. The fertilized egg is called the zygote and has 1 set of chromosomes from each parent. The zygote produces somatic cells by mitosis and then develops into an adult. At sexual maturity, the ovaries and testes produce haploid gametes. Gametes are the only types of human cells produced by meiosis rather than mitosis. Meiosis results in 1 set of chromosomes in each gamete. Fertilization and meiosis alternate in sexual life cycles to maintain chromosome number. Okay, so here we have a female which has ovaries which will produce eggs through meiosis. We have a male who has testes which will produce sperm through meiosis. The sperm fertilizes the egg to become a diploid zygote, and then that zygote will grow and develop through mitosis into an adult individual. The alternation of meiosis and fertilization is common to all organisms that reproduce sexually. The three main types of sexual life cycles differ in the timing of meiosis and fertilization. Gametes are the only haploid cells in animals. They are produced by meiosis and undergo no further cell division before fertilization. Gametes fuse to form a diploid zygote that divides by mitosis to develop into a multicellular organism. So that's pretty much the same as the human life cycle. So here's an animal, diploid multicellular organism, which is what we are. Plants and some algae exhibit an alternation of generations. This life cycle includes both a diploid and haploid multicellular stage. The diploid organism, called the sporophyte, makes haploid spores by meiosis. Each spore grows by mitosis into a haploid organism called a gametophyte. A gametophyte makes haploid gametes by mitosis. Fertilization of gametes results in a diploid sporophyte. Okay, so remember in angiosperms, the diploid sporophyte is the plant that you see, and the haploid multicellular organism is housed within the diploid sporophyte. So here we have the diploid multicellular sporophyte, which has a chromosome number of 2n, which will undergo meiosis to create haploid spores, and then those haploid spores, which are right here, will grow through mitosis to become the haploid multicellular organism, or the gametophyte. Okay, so it's one set of chromosomes, therefore it's haploid, and it's a gametophyte. So they alternate between gametophytes and sporophytes. So then that gametophyte will produce gametes through mitosis, and then fertilization will occur. Sperm fertilizes the egg to create the diploid zygote, which will then grow into the diploid multicellular organism, or the sporophyte. In most fungi and some protists, the only diploid stage is the single-celled zygote. There is no multicellular diploid stage. The zygote produces haploid cells by meiosis. Each haploid cell grows by mitosis into a haploid multicellular organism. The haploid adult produces gametes by mitosis. Okay, so this diploid zygote, this is the only diploid stage of this life cycle. So the diploid zygote divides by meiosis to create haploid cells, and then that will grow into the haploid unicellular or multicellular organism through mitosis. And then through mitosis, gametes will be produced. It has to be mitosis because it's already a haploid number of chromosomes. So through mitosis, the organism produces gametes, which then the sperm fertilizes the egg to become the zygote. Depending on the type of life cycle, either haploid or diploid cells can divide by mitosis. However, only diploid cells can undergo meiosis. In all three life cycles, the halving and doubling of chromosomes contribute to genetic variation in offspring. Like mitosis, meiosis is preceded by the replication of chromosomes. So right before mitosis and right before meiosis, the chromosomes get replicated. Meiosis takes place in two sets of cell divisions called meiosis 1 and meiosis 2. Two cell divisions result in 4 daughter cells rather than the 2 daughter cells in mitosis. Each daughter cell has only half as many chromosomes as the parent, so it's a reduction division. For a single pair of homologous chromosomes in a diploid cell, both members of a pair are duplicated. The resulting sister chromatids are closely associated along their lengths. Homologs may have different versions of genes, each called an allele. For example, a gene might be eye color. Different alleles would be brown eyes, blue eyes, green eyes, and so on. Homologs are not associated in any obvious way except during meiosis. Okay, so here we have a pair of homologous chromosomes. After the S phase interphase, they become duplicated chromosomes. After meiosis 1, the pairs separate, and after meiosis 2, the sister chromatids are ripped apart at their centromeres. So we end up with haploid cells. Meiosis halves the total number of chromosomes very specifically. It reduces the number of sets from 2 to 1, with each daughter cell receiving 1 set of chromosomes. In the first meiotic division, homologous pairs of chromosomes pair and separate. In the second meiotic division, sister chromatids of each chromosome separate. 4 new haploid cells are produced as a result. Okay, prophase 1. Prophase 1 typically occupies more than 90% of the time required for meiosis. This is when crossing over occurs, so chromosomes begin to condense. In synapsis, homologous chromosomes loosely pair up, aligned gene by gene. This allows crossing over, where non-sister chromatids exchange DNA segments. Each homologous pair has one or more X-shaped regions called chiasmata. Chiasmata exist at points where crossing over has occurred, so crossing over occurs at a very high frequency. Metaphase 1, this is when the homologous pairs line up at the metaphase plate. We call them tetrads because you have a pair of homologous chromosomes, and each pair is duplicated. So really you have 4 copies of that same chromosome. They're just going to have different alleles because one's male and one's female. One comes from mom, one comes from dad. Microtubules from one polar are attached to the kinetochore of one chromosome of each tetrad. Microtubules from the other polar are attached to the kinetochore of the other chromosome. So this will allow these microtubules or spindle fibers to separate the pairs of homologous chromosomes, which is what happens in anaphase 1. Pairs of homologous chromosomes separate. One chromosome moves towards each pole, guided by the spindle apparatus. Sister chromatids remain attached to the centromere and move as one unit toward the pole. Telophase 1 in cytokinesis. In the beginning of telophase 1, each half of the cell has a haploid set of chromosomes. Each chromosome still consists of 2 sister chromatids. Cytokinesis usually occurs simultaneously, forming 2 haploid daughter cells. Now remember, after meiosis 1, you're already down to a haploid set of chromosomes because 23 chromosomes go to one cell, 23 duplicated chromosomes go to one cell, and 23 duplicated chromosomes go to the other cell. Okay? So you went from a diploid cell that has 46 to 2 haploid cells that have 23 duplicated chromosomes. In animal cell, a cleavage fiber forms. In plant cell, a cell plate forms. That cell plate will become the cell wall of a plant cell. No chromosome replication occurs between the end of meiosis 1 and the beginning of meiosis 2 because the chromosomes are already replicated. So it was just the pairs that got separated. Division in meiosis 2 also occurs in 4 phases: prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Meiosis 2 is extremely similar to meiosis. The only difference is that you're starting with 23 chromosomes, 23 duplicated chromosomes, instead of 46 duplicated chromosomes. In prophase 2, a spindle apparatus forms. In late prophase 2, chromosomes move toward the metaphase plate. In metaphase 2, the sister chromatids are arranged at the metaphase plate. Because of crossing over in meiosis 1, the 2 sister chromatids of each chromosome are no longer genetically identical. So crossing over leads to a lot of genetic variation in gametes. The kinetochores of sister chromatids attach to microtubules extending from opposite poles. In anaphase 2, the sister chromatids separate. The sister chromatids of each chromosome now move as 2 newly individual chromosomes toward opposite poles. In telophase 2, the chromosomes arrive at opposite poles, nuclei reform, and the chromosomes begin decondensing. At the end of meiosis, there are 4 daughter cells, each with a haploid set of unduplicated chromosomes. Each daughter cell is genetically distinct from the others and from the parent cell, so you have a lot of genetic variation in the gametes. Comparing mitosis and meiosis. Mitosis conserves the number of chromosome sets, producing cells that are genetically identical to the parent cell. So mitosis, you start with a 2N cell, you end with a 2N cell, genetically identical. In humans, you go from 46 chromosomes in the parent cell to 46 chromosomes in the daughter cell. Meiosis reduces the chromosome number from 2 to 1. So in meiosis, you're starting with a parent cell that has a 2N number of chromosomes, or 46 in humans. and you end up with haploid number of chromosomes in the 4 daughter cells, so 23 in humans, or haploid as a general term. And these gametes are not genetically identical to the parent cells. Meiosis includes 2 divisions after replication, each with specific stages. Mitosis, you only have 1 division after replication. 3 events are unique to meiosis, and all 3 occur in meiosis 1. Synapsis and crossing over, which occurs in prophase 1. Homologous chromosomes physically connect and exchange genetic information. Homologous pairs at the metaphase plate. Homologous pairs of chromosomes come together in metaphase 1, so they line up as pairs, not as individual chromosomes. And then separation of these homologs, or separation of these homologous pairs during anaphase 1, which leads to the cells after the first round of division being haploid. So this is a nice side-by-side comparison that you should probably read over. Okay, this is a good chart that you should read over to review the differences between mitosis and meiosis. Sister chromatid cohesion keeps sister chromatids of a single chromosome attached during meiosis and mitosis. Protein complexes called cohesins are responsible for this cohesion. In mitosis, cohesins are cleaved at the end of metaphase. In meiosis, cohesins are cleaved along the chromosome arms in anaphase 1, separation of homologs, and at the centromeres in anaphase 2, which allows the separation of sister chromatids. Meiosis is called the reductional division because it halves the number of chromosome sets per cell from diploid to haploid. Meiosis 2 is called the equational division because the haploid cells divide to produce haploid daughter cells. Okay. The mechanism of sister chromatid separation in meiosis 2 is identical to that in mitosis. Mutations are the original source of genetic variation. Mutations create different versions of genes called alleles, and reshuffling of alleles during sexual reproduction produces genetic variation. The behavior of chromosomes during meiosis and fertilization is responsible. The behavior of chromosomes during meiosis and fertilization is responsible for most of the variation that arises in each generation. So there are 3 mechanisms that contribute to this genetic variation. The independent assortment of chromosomes. So the homologous pairs line up randomly. They line up chromosome number 1 with chromosome number 1, chromosome number 2 with chromosome number 2, but they line up randomly. So in other words, on one side, chromosome 1 could be the parental chromosome, and on the right side, it could be the maternal chromosome, or vice versa. Think of that 46 times, okay? Well, actually, yeah, 23 times. So 23 pairs of chromosomes lining up randomly at the metaphase plate, and they sort independently. So you have many different variations in your gametes with regard to which maternal and paternal chromosomes end up in which cell. And then you have crossing over, the exchange of genetic information between homologous chromosomes, and then you have random fertilization, which just means that any sperm can fertilize any eggs. Homologous pairs of chromosomes orient randomly at metaphase 1 of meiosis. In independent assortment, each pair of chromosomes sorts maternal and paternal homologs into daughter cells independently of the other pairs. The number of combinations possible when chromosomes assort independently into gametes is 2 to the n, where n is the haploid number. So in humans, that's 23, so there's 8 million possible combinations of chromosomes. And again, this is just showing how the homologous pairs orient randomly at the metaphase plate. So if we have just 2 pairs of homologous chromosomes, so 2n equals 4, then what we would have here is we could have the male one lining up on this side and the male one also lining up on this side, so chromosome 1, chromosome 2. Or we could have the male lining up on this side and the second male lining up on the other side. So that leads to variation in the gametes. So think of that with 23 pairs of chromosomes lined up. How many possible combinations that you could have. Crossing over produces recombinant chromosomes, which combine DNA inherited from each parent. Crossing over begins very early in prophase 1 as homologous chromosomes pair up gene by gene. In crossing over, homologous portions of two non-sister chromatids trade places. Crossing over contributes to genetic variation by combining DNA, producing chromosomes with new combinations of maternal and paternal alleles. So here we have the homologous pair of chromosomes coming together during synapsis. We have crossing over that's occurring. We have chiasma, plural chiasmata, where these little X-shaped regions where crossing over has occurred. And then here's the result of your crossing over. You have homologous chromosomes that have pieces from the other homologous chromosome. As a result, there is even more genetic variation in your daughter cells. So here we have even more genetic variation because these have both paternal and maternal genes on them. Random fertilization adds to genetic variation because any sperm can fuse with any egg. The fusion of two gametes, each with 8.4 million possible chromosome combinations from independent assortment, produces a zygote with any of about 70 trillion diploid combinations. Crossing over adds even more variation to that. Each zygote has a unique genetic identity. Natural selection results in the accumulation of genetic variations favored by the environment. Sexual reproduction contributes to the genetic variation in a population, which originates from mutations. Asexual reproduction is less expensive than sexual reproduction. So in asexual reproduction, you don't have to go out and actively seek a mate. So that's one advantage. So in a relatively stable and unchanging environment, asexual reproduction would actually be beneficial. But since we have an ever-changing environment, sexual reproduction is more favored because in a changing environment, if the environment changes, then the population is going to want to have genetic variation in order to be able to adapt to the change in the environment. So overall, genetic variation is evolutionarily advantageous.